Very good morning, students. Welcome to the online video lesson. Today's topic is plant physiology, mineral nutrition. Mineral nutrition in plants. As you all know, plants take up mineral elements in the form of inorganic ions. These inorganic ions are available in the form of minerals in the soil. So, the nutrition of inorganic ions of plants is called mineral nutrition. So the plants need at least 16 elements for nutrition, which is suggested by the scientist known as Arnon. These elements are known as essential elements. These may be required in small amount or in large amount. So the elements such as carbon, hydrogen, oxygen, nitrogen, phosphorus, potassium, calcium, magnesium and sulfur. These elements are required in more concentration. These particular elements required in large concentrations. So these are called as macro elements. In the same way, some of the elements which are required in small concentrations, which are called as micro elements, such as iron, zinc, copper, manganese, molybdenum, boron and chlorine. So the growth of plants in a nutrient medium is called hydroponics. This was introduced by Grillrich. This led to analyze the deficiency symptoms in plants when these was a deficiency of the element. So whatever may be the elements which are not uh, supplied to the plants or which are not available to the plants, these particular nutrients, deficiency of these particular nutrients make or elements make the plant to suffer from several diseases or abnormalities. So when we move deep into the topic, this is the diagram which represents whatever may be the requirements of the plants in the form of nutrients. So nutrients initially they are absorbed by these roots along with the water. So ions are being absorbed from these particular roots to root hairs, and again some of the minerals are developed in the aerial parts of the plant, that is stem and leaves, during photosynthesis. Okay, so the carbohydrates, as you all know, the glucose is synthesized or starch is synthesized during photosynthesis by utilizing the sunlight. So many elements are present in the complex forms, but they are required in simple forms also. Some of the simple forms can also be absorbed through air, through stomata, while the gaseous exchange will take place. So so many aspects are there, so many metabolic processes are there, where the plant utilizes these micro and macro elements in order to synthesize the food material or transpire or respire or respiration process. Okay, so when we see initially the mineral elements which are divided into two forms, that is essential and non-essential forms. So essential elements are those which are necessary for normal growth and development, and non-essential elements which are not involved in the growth and development, but in other aspects they are essential. So basically there are two types, essential and non-essential. And when we see the criteria for essentiality, according to Epstein in 1972, what is an essential element? An element is essential if the plant cannot complete its life cycle, that is from viable seed, in the absence of the element. So whatever may be the deficiency of the element which leads to the abnormality. Okay, so an element is essential if it forms part of any molecule or constituent of the plant, is itself essential for the plant. For instance, nitrogen in proteins and magnesium in chlorophyll. So the some of the elements they are in the complex forms, whether they may be present in the molecule itself within the plant. Sometimes it is taken individually from the external sources. So this is the essentiality criteria can be seen for essentiality. When we see the essential elements, specifically, when we see the essential elements, they are divided into macronutrients and micronutrients. So macronutrients are the elements which are considered as the major elements required in large amount, more than 10 millimolecules per kg of dry matter. Whereas the micro elements which are treated as minor elements or trace elements, which requires in very small amounts, that is less than 10 millimoles per kg uh, dry matter. Okay, so these are macro and micronutrients. When we see in detail, when we see the functions of essential elements, so the functions can be categorized. The constituents of protoplasm of cell wall, it may be one function. Influence on the osmotic pressure of plant cells, it is also one of the functions or essentiality of the elements. Next, antagonistic of balancing function against toxic elements. So this is the response of the plants towards the external mm, environment. And one of the function is a catalytic function, whatever may be the metabolic activities or reactions which are taking place inside the plants are determined by these particular elements. So 17 elements are deemed to be essential for plant growth and development. Out of that, some are classified under macro elements, some are under micro elements. So macro elements are, as we already discussed, that hydrogen, carbon, oxygen, nitrogen, uh, potassium, calcium, magnesium, phosphorus, and sulfur. They are treated as macronutrients, and also we need to understand they are in their availability form. Okay, whatever may be the available forms for the plant, these particular macronutrients which are present in complex structure, okay, whatever molecular forms, we need to first know about it. So where this hydrogen is present and how it is supplied to the plant. Hydrogen is generally present in the form of water. So concentration in dry matter, millimoles per kg, when we see 60,000 millimolecules moles per kg. And when we see the carbon source, the source of carbon is carbon dioxide, which is available in the air. Okay, so it is 40,000 millimoles per kg required amount concentration in dry matter when we observe. Okay, so when we see the oxygen is in the form of carbon dioxide, or maybe pure form oxygen is which is present in the air. Okay, it is 30,000 uh, concentration millimoles per kg concentration in dry matter. In the same way, whatever may be the concentration, we will see one by one. And nitrogen, which is present in the form of nitrates and nitrites, it may be 1000 millimoles per kg in dry weight. In the same way, potassium, they are individually, they are available to the plants in the ionic forms. In the same way, calcium ions are also individually, they are available in the form of ions. That is, it is, when we see the molar concentration, it is 125 millimoles per kg. In the same way, magnesium magnesium ions are present, 80 millimolecules per kg. In the same way, phosphorus, they are available in phosphoric acid forms, that is 60 millimoles. In the same way, sulfur, when we see the uh, sulfur oxide forms, and having 30 millimoles per kg. So these are some of the macro elements. When we come to the micro elements, so chlorine, boron, iron, manganese, zinc, copper, nickel, and molybdenum are treated as micro elements. So these micro elements, for example, chlorine, are readily available in the form of chlorine ions, that is 3.0 millimoles per kg dry weight. And boron, when we see, it is it is found in the form of oxide forms. It has a weight around 2.0. And iron, it is also available in the free forms, that is uh, ferrous forms. And again, when we see the manganese, it is also readily available in the form of ions, that is 1.0 millimoles per kg. In the same way, zinc, copper, nickel, molybdenum, these are all available in the oxide and ionic form, that is calcium 1, and least amount, when we see the weight, that is nickel and molybdenum, they are required in very small amount, they are present in very small amount when we see the dry weight of the plant body. So these are the concentrations of various concentration of macro and micro elements. So when we move to the first major macro element, that is nitrogen, which is required in large amount. So nitrogen is very much essential for the plant to in all metabolic activities. So when we see the functions and deficiency symptoms separately, what is the function of nitrogen in plants? So nitrogen function is, it is a constituent of amino acids, amides, proteins, nucleic acids, nucleotides, coenzymes, and hexoamides, etc. So these all the metabolic activities are being catalyzed by this particular nitrogen element. So in all complex forms, this nitrogen is present. If the deficiency of nitrogen is present in the plant, it shows a yellowing, that is chlorosis of leaves, yellowing of veins and leaves. In the same way, petioles of leaf veins colored due to deposition of anthocyanin. So plant growth is stunted. So due to the deficiency symptoms of nitrogen, you can see all these symptoms in the plant. In the same way, phosphorus, when we see the function, it is a component of sugar phosphates, that is nucleic acids. As you all know, phosphorus is present in the form of phosphoric acid in DNA backbone. And nucleotides, coenzymes, phospholipids, phytic acid, etc., has a key role in reaction that involves ATP. So whatever may be the energy which is being stored in the cell is involved by this element phosphorus. When the phosphorus deficiency symptoms, when we observe premature leaf fall will be seen in, and dead necrotic aura development on fruits and leaves. So some of the spots are developed on the fruits and leaves, and leaves turn dark to blue green in color. So this is the symptoms of deficiency of phosphorus. When we see the sulfur function, the component of it is one of the component of cysteine, methionine, and proteins. It is a constituent of lipoic acid, coenzyme A thiamine, and also pyrophosphate, glutathione, biotin, adenosine 5-phosphosulfate, and 3-phosphoadenosine. So these are the functions of sulfur. And deficiency symptoms of sulfur, if the sulfur is present in low amount, chlorosis of leaves can be observed, margin and tips of leaves roll inward, stem become hard due to development of sclerenchyma. So these are the deficiency symptoms. So when we move to the calcium, the function of calcium, it is the chief constituent of the middle lamella of cell wall. As you all know, it lays cell wall in between um, the daughter cells which are formed after uh, telophase stage, uh, cytokinesis stage. When we observe, it is required as a factor by some enzymes involved in the hydrolysis of ATP and phospholipids, and acts as a second messenger in metabolic regulations. Um, and also, when we see the calcium which is present in the cell walls, which, in the middle lamella, okay, it is very strong. As you all know, the cell wall of the plant cell is very much tough due to the presence of cellulose and other segment, but this calcium also deposited in the middle lamella and gives some flexibility or strongness to this particular cell wall, or toughness to the cell wall. Okay, so deficiency symptoms: disintegration of growing meristematic regions of roots, chlorosis along leaf margins, malformations of younger leaves can be seen due to the deficiency of calcium. So magnesium, when we see, it is required by many enzymes involved in phosphate transfer. Constituent of the chlorophyll molecule play role in binding ribosomal particles during protein synthesis. So when we come to deficiency symptoms of magnesium, chlorosis of leaves and dead and necrotic patches develop on leaves can be seen in the deficiency symptoms of magnesium. When we observe the potassium functions, it is required as a cofactor for more than 40 enzymes. It is principal cation in establishing cell turgor and maintaining cell electroneutrality, play an important role in stomatal movement also. So that when we see the deficiency symptoms, deficiency of potassium causes mottled chlorosis of leaves. Uh, Sometimes dead necrotic patches develop at tips and margins of leaves. These are the symptoms, deficiency symptoms of potassium. When we see the iron function, it is the chief constituent of cytochromes and non-heme iron proteins involved in photosynthesis, nitrogen fixation, and respiration. So deficiency symptoms of irons leads to the formation of a rapid chlorosis of leaves. So iron is an important aspect of plant constituent. Nutrition is required. Okay. Next, manganese. When you see the manganese function, manganese is required for activity of some dehydrogenases, decarboxylases, kinases, oxidases, and peroxidases involved with other cation activated enzymes and photosynthetic oxygen evolution. So deficiency symptoms of manganese leads to a rapid chlorosis and necrotic spots of leaves. So these are the symptoms. When we observe the copper functions, copper is the component of ascorbic acid oxidase, tyrosinase, monoamine oxidase, uricase, cytochrome oxidase, phenolase, uh, lactase, and plastocyanin. So these are the chief constituents of components of these particular substrates. And when we see the deficiency symptoms of copper, due to the deficiency of copper, necrosis of tips of leaves are observed. The dieback of citrus and reclamation disease of cereals and leguminous plants are observed due to copper deficiency. In the same way, when we see the zinc functions, it is the constituent of alcohol dehydrogenase, glutamic dehydrogenase, carbonic anhydrase, etc. Deficiency symptoms of zinc leads to chlorosis of older leaves. Mottle leaf diseases of apple, citrus, walnut, etc. are seen. uh, due to the deficiency of zinc. Okay, so one more element, that is boron. When we see the functions, so it is in the complexes with mannitol, mannan, polymannonic acid, and other constituents of cell walls involved in cell elongation and nucleic acid metabolism. The deficiency of boron leads to the death of shoot tip, that is flower formation suppressed, root growth stunted, leaves become coppery in texture. So these are the symptoms of deficiency of boron. Next, molybdenum functions. When we see, it is a constituent of nitrogenase, nitrate reductase, and xanthine dehydrogenase enzymes. And deficiency symptoms of molybdenum are seen with chlorotic intervenal mottling of older leaves, and also flower formation inhibited. Whiptal disease in cauliflower plants can also be seen due to the disease deficiency disease of molybdenum. When we see the chlorine functions, it is required for the photosynthetic reactions involved in oxygen evolution. In the same way, when we see the deficiency symptoms of chlorine, which leads to wilting of leaf tips followed by chlorosis and necrosis. Leaves show reduced growth, the roots become stunted in length but thickened near the tips. So these are the deficiency symptoms. When we see the nickel, it is required in a small amount, very small amount when we see the dry weight of the plant. When we see the necrotic function, it is a constituent of urease enzyme in end-fixing bacteria. It is a constituent of hydrogenases also. So deficiency symptoms leads to accumulation of urea in leaves. Necrosis of leaf tips occurs due to the deficiency of nickel. So so beneficial elements, in addition to the 17 essential elements, some plants appear to have additional requirements. However, because these have not been shown to be requirements of higher plants, generally they are excluded from the list of essential elements. They are referred to instead as beneficial elements. The definition of beneficial currently applies primarily to sodium, silicon, selenium, and cobalt. So these are the beneficial elements in addition to 7 essential elements. When we see the sodium function, it is generally essential as a micronutrient for plants utilizing specifically the C4 photosynthetic pathways, but not for most C3 plants. It may be related to transport of pyruvate between bundle sheath cells and mesophyll cells. So this is important in the C4 cycle. When we see the deficiency symptoms of this particular sodium, it leads to plant growth in a depleted solution, showed reduced growth, chlorosis, yellowing due to loss of chlorophyll, and necrosis. Dead tissues of the leaves are observed. When we see the silicon functions, it is beneficial to grasses, accumulates in the cell walls, especially of epidermal cells, and may play a role in uh, finding of fungal infections or preventing lodging, a condition in which stems are bent over by the heavy winds or rain. So these are the functions of silicon. The same way, deficiency symptoms, when we observe, they are more susceptible for fungal infections and lodging. Okay, so deficiency of silicon leads to fungal infections. When we see cobalt function, cobalt is essential for growth of legumes. In this case, the requirements can be traced to the needs of the nitrogen fixing bacterium rather than the host plant. When legumes are provided with fixed nitrogen such as nitrate, a cobalt requirement cannot be demonstrated. Deficiency symptoms of cobalts reduce the legume bacterial association. Okay, so these are the symptoms of cobalt deficiency. When we see the selenium, when we see the functions, generally toxic of most plants, certain members of the legumes genus such as astragalus, milkweed or poison wedge tolerate high concentration of selenium up to 0.5 percentage dry weight. High selenium content in these plants causes a sickness known as alkali poisoning or blind staggers in grazing animals. So, no, not much deficiency symptoms are seen. Uh, only the uh, amount, if the quantity increases, then only some of the symptoms are seen in the plants, but not the deficiency. Okay, so it is required in small amounts. Next, when we see the references for this mineral nutrition, Plant Physiology by Tyes and Ziegler, third edition you can follow. Introduction to Plant Physiology by William G. Hopkins and Norman, P.A. Hunter, fourth edition. You can refer these references in order to understand the mineral nutrition. So, minerals play an important role. As you all know, animals consume food, different types of foods in order to get different kinds of nutrients, whether it may be carbohydrates, proteins, vitamins, so and minerals. But plants, they doesn't have any of this particular separate digestive system like animals. They need to absorb the ions or water, and the substances in the form of gases, elements in the form of gases. So they have separate mechanism to synthesize their food material. So these materials are required by the plants, okay, naturally. If we supply these particular means whatever may be the symptoms which we are studied now can be clearly understood by tissue culture techniques by supplying various nutrients in order to understand the mineral nutrition. As we all know, in tissue culture techniques we are supplying the nutrient medium, okay, in order to raise different structures of the plants from the callus. So where we can understand it very clearly, uh, rather than supplying nutrients for the living plant in the open air. Okay, so naturally, which is available naturally, it will take time to show different types of symptoms. So these can be understood better in the laboratory conditions. Okay, so we can test all these mineral nutrients, and their functions and deficiency symptoms in the laboratory itself. So these tissue cultures plays an important role in understanding the mineral nutrition also. So this is an important topic in physiology, plant physiology, apart from water relations. So mineral nutrition is very important in plants in their growth and development. So try to make notes from these slides. It is very much enough for you to make your notes. If you want additional information, you can refer the following books, and try to write the notes and make an assignment of this particular mineral nutrition. It is very important, and try to make a project work also. Okay, various plant diseases due to deficiency symptoms and requirements you can measure, and micro elements you can make it as a project also. So try to make one project from this plant physiology, especially mineral nutrition in plants. Okay, hope you understand the class well. Try to uh, write the notes from these particular slides. Hope you understand the class well. Thank you.